Good morning, dear students. Good morning.
Good morning, ma'am.
I think by October 3rd week, we'll complete our portions. And October last week, I think we planned of giving you one practical exam. University model practical exam. October 25th in the week. Okay. എട്ട് പത്തായിട്ടും 60 പേരൊക്കെയാണ് ക്ലാസ്സിൽ കയറിയേക്കുന്നത്. Translation and repeat for your exam. It's very important. It will be asked as an essay question for you. I think what I am sharing is a picture from Harper, and the diagram might be very useful for you. It is self-explanatory. Self-explanatory. If you draw the three figures, that is initiation step, elongation, termination, moon figures on. If you draw the figures, it will be more than enough to get more than 90% for the essay. Mark if you draw the picture. So try to take down the figure also. ക്ലാസ്സിൽ 10-70 കുട്ടികളാണ് ഉള്ളത്. വെരി പുവർ അറ്റൻഡൻസ്. എന്താണ് അറിയില്ല എന്തുവാണെന്ന്. Okay. So last class we are dealing with translation. I told you translation mainly there are 3 major steps in translation and a preparatory step during which the amino acids are being activated. Amino acids are being activated. How it is being activated? Amino acids will combine with ATP. 2 high energy phosphate is being utilized, forming amino acid AMP, and this amino acid AMP get attached to tRNA, the specific tRNA. having the specific anticodon. Based on the anticodon and the codon of mRNA, specific amino acids get attached to the CCA 3-hydroxy group of tRNA. Understand? Forming aminoacyl tRNA. Thus, aminoacyl tRNAs are getting formed. Then, coming to the initiation step. So, during initiation, what happens? ഞാൻ repeat ചെയ്ത് പറയുവാണ്. Repeat ചെയ്യുന്നെന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ it's very during the initiation step. First step is dissociation of ribosome into 40S and 60S subunit. Dissociation of ribosome into 40S and 60S subunit. So that is the first step, dissociation. Dissociation of ribosome into, so the 80S ribosome is dissociated into 40S and 60S subunit, to which the eukaryotic initiation factor, it is represented like this. Eukaryotic initiation factor 3 and 1A will bind to the 40S subunit, preventing its reassociation with 60S subunit. And to this 40S subunit, so the next step is formation of 43S pre-initiation complex. Formation of 43S pre-initiation complex. So to this 40S subunit, the ternary complex composed of methionine tRNA and factor 2 along with GTP will bind, forming the 43S pre-initiation complex. Formation of 43S pre-initiation complex. Now comes the second step is, the third step is formation of 48S initiation complex, to which this mRNA has to get attached. This mRNA, the complex structure of mRNA has to be removed. The factor 4F, eukaryotic initiation factor 4F, it is composed of 4E, 4G, 4A complex. Understand? 4F is composed of 4E, 4G, 4A complex. It will bind to the cap region. It will bind to the cap region. Then the factors, eukaryotic, other initiation factors, eukaryotic initiation factor 4A and 4B will bind to the mRNA and will remove the complex secondary structure of mRNA. Now the mRNA is ready to attach to the 43S pre-initiation complex, forming the 48S initiation complex. The next step is formation of 48S initiation complex. Understand? So this ribosome, the 43S complex will read the mRNA. It will read the mRNA and it will attach to the AUG. Understand? It will attach to AUG. See, you know, it has got the methionine. Understand? So it get attached to the initiator codon. Initiator codon is being designated by the sequence Shine-Dalgarno Cossacks sequence. Cossacks is a Cossack sequence which determine where the initiator codon is. Understand? With the and it will bind to the initiator codon, forming the 48S initiation complex. 48S initiation complex. So the next step is, which is the next step? S, which is the next step? One minute. ഇതൊന്ന് മാറ്റട്ടെ. Sorry. Yes. Formation of 48S initiation complex. So this the 48S initiation complex is, next step will be the formation of 80S initiation complex. So what happens? The eukaryotic initiation factor 5 will come. Eukaryotic initiation factor 5 will come. Understand? When eukaryotic initiation factor attaches to 48S initiation complex, what happens? The other eukaryotic initiation factor 3, 1A and 2 are removed. 3, 1A and 2 are removed. And so the 60S subunit can bind to the 48S initiation complex. Understand? So the 60S subunit can bind to the 48S initiation complex, forming 80S initiation complex. Understand? So you can see that in the ribosome, there are ribosomes, there are 3 sites. Understand? Mainly in a ribosome, there are 3 sites. They are the P site, mainly the P site, A site, and E site. And at this P site, the initiator, the AUG carrying or methionine tRNA is attached. Understand? The methionine carrying tRNA is attached at the P site. So the A site is free. Now it enters into this complex will enter into the elongation phase. It then enters into the elongation phase. I think we have covered up to this much. Hope most of you have taken down the figure. വരച്ചെടുത്തിട്ടുണ്ടോ?
Yes, ma'am.
Yes. Yes. Now moving on to the elongation phase. So the A site is free. Coming to the elongation phase. See, during the elongation phase, we can see this site is free. So here we can see at the start, methionine, methionine carrying tRNA. So the A site is free. If at this region, if the codon is UUU, if the codon is UUU, so which amino acid will come? Which tRNA will come? The phenylalanine carrying tRNA. Understand? The phenylalanine carrying tRNA will come and attach at the A site. So the first step under the first step in elongation is binding of aminoacyl tRNA to A site. So the first step in elongation is binding of aminoacyl tRNA to aminoacyl tRNA to A site. Understand? So based on the codon in A site, the corresponding the specific tRNA will come and attach. It requires which all factors? It requires the eukaryotic elongation factor. It requires the eukaryotic elongation factor 1 alpha. Understand? It requires the eukaryotic elongation factor 1 alpha along with eukaryotic elongation factor 1 alpha and GTP, the corresponding the specific amino acyl tRNA will come and occupy the A site. So that is the first step in elongation. That is binding of amino acyl tRNA to A site. Understand? The next step is peptide bond formation. The next step in elongation is peptide bond formation. Peptide bond formation. Understand? So what happens in this step? So so in the next step, the amino acid that is attached at the P site will form a peptide bond with the amino acid occupying the A site. See, if here it told you here it is methionine, no? Methionine. So here it is phenylalanine. So the methionine will form a peptide bond with phenylalanine. Methionine will form a peptide bond with phenylalanine. Understand? Forming phenylalanine ഇവിടെ methionine. Understand? Will form a peptide bond with the amino acid occupying the A site. The enzyme is peptidyl transferase. Peptidyl transferase. We are dealing with eukaryotic translation process now. So the peptidyl transferase is having 28S ribosomal subunit in it, and it is an example for ribozyme. What is the difference between ribozyme and enzymes? Ribozymes can be asked as 1 mark question for you. What are ribozymes? Ribozymes are enzymes having RNA component in it. Examples are peptidyl transferase, RNase P, RNase H, etc. are examples for ribozymes. So it will, with the help of peptidyl transferase, a peptide bond is formed. So that is the second step, peptide bond formation. So what will be the amino acid at the P site? The tRNA at the P site will be vacant. No, it is empty. It has formed a peptide bond with the amino acid occupying the P site. So it is empty, and this will be removed to the exit site. It moves out of ribosomes. So now the P site is free. Understand? Now the P site is free. So the P site is empty. P site is empty. So the A site is carrying the peptide chain carrying tRNA. Understand? So the P site is free. So the third step in elongation is translocation process. The third step is translocation. Translocation. So what happens? So the tRNA occupying the A site occupies the P site. Is translocated to the P site. And this step requires energy. This step requires GTP and eukaryotic elongation factor 2. Eukaryotic elongation factor 2. So with the help of eukaryotic elongation factor 2 and GTP, the peptidyl tRNA translocates to the P site. Understand? Translocates to the P site. Now the A site is free. Now the A site is free. Understand? So the A site is free. Understand? So it translocates to the P site. Now the A site is free. And based on the codon in at the A site, the specific tRNA will come and attach, and the process goes on. Understand? So that is the elongation phase. Under elongation phase, there are 3 steps. Which are the 3 steps? One is the binding. First step is binding of aminoacyl tRNA to A site. The second step is peptide bond formation with the help of peptidyl transferase enzyme. This peptidyl transferase enzyme is having 28S ribosomal subunit in it. And the third step is translocation. That is the peptidyl tRNA occupies the P site, and A site is free to carry on the next step, a step in elongation. Understand? So how many molecules of GTP are being utilized? 2 molecules of GTP. 2 high energy phosphate are being utilized in this step. Understand? So That is about elongation. Hope you might have taken down the figure. വരച്ചെടുത്തായിരുന്നോ കുട്ടികളെ? എസ് അതുൽ കൃഷ്ണൻ
Yes, ma'am.
ക്ലാസ്സിലുണ്ടോ?
ഉണ്ടല്ലോ.
Were you able to take down the figure?
Ma'am.
Okay. So that is about elongation. ഇർഫാൻ ക്ലാസ്സിലുണ്ടോ? ഇർഫാൻ ക്ലാസ്സിൽ കയറിയിട്ടുണ്ടോ? റോൾ നമ്പർ 4 ആദിത്യ അശോക്.
Yes, ma'am.
Are you in the class? Can
Yes,
you
ma'am,
explain
in
the
the class.
elongation, please? Aditya?
Yes, ma'am.
Yes.
Ma'am,
Yes. I will show you the figure. Arnolo, what happens? Which are the 3 major steps in elongation? Arnolo.
ma'am, identification of the codon.
Binding of ഈ 3
Binding
സ്റ്റെപ്പിന്റെ
of
പേരുകൾ
the-
അത് തന്നെ വേണം കേട്ടോ. ബൈൻഡിങ് ഓഫ് അമിനോ അസൈൽ tRNA to A site. Binding of amino acyl tRNA to A site. Based on the codon in A site, the specific amino acyl tRNA will occupy the A site. Which is the second step.
Yes, ma'am.
So which are the factors that are needed for binding the tRNA to A site? Which are the factors that are needed? Yes, Aditya.
Ma'am, the anticodon, the codon, reading of the anticodon.
ഏത് ഫാക്ടേഴ്സ് ഒക്കെയാണ് വേണ്ടത് to bind amino acid tRNA to A site?
Eukaryotic elongation factor 1 alpha.
And GTP.
GTP.
അല്ലേ?
Yes, ma'am.
Along with this, it will come and bind to the A site. So next, which is the second step, Aditya?
Ma'am, peptide bond formation.
Yes, peptide bond formation. So what happens with the which is the enzyme?
Peptidyl transfer.
Peptidyl transferase. Peptidyl transferase. And the third step is
Translocation.
translocation. ആദിത്യ ക്ലാസ്സിൽ ശ്രദ്ധിക്കുന്നുണ്ടോ?
ഉണ്ട് ma'am.
ജിഷ്ണ ഉണ്ടോ ക്ലാസ്സിൽ? ജിഷ്ണ, ആർ യു ഇൻ ദ ക്ലാസ്? ജിഷ്ണ വി ക്ലാസ്സിൽ കയറിയിട്ടില്ല. ജിഷ്ണ, പാർവതി ദാസ് ക്ലാസ്സിലുണ്ടോ? പാർവതി, are you in the class? Yes. Yes, which are the 3 major steps in elongation? Yes, Parvati, which are the 3 major steps in elongation?
Binding of amino acid tRNA, peptide bond
Then-
formation, and translocation.
Yes. Now, moving on. Yes, Parvati. Now, moving on to the termination phase. So, the next step is termination. So, what happens? We'll see. See, during the termination, we can see if a stop codon come at this region. Which are the stop codon? UAA, UAG, and UGA. So what happens when a stop codon, let UAA come at this region? So there is no tRNA, no amino acid tRNA to recognize this site. Understand? So what happens? Releasing factor 1, RF1 and RF3 along with the GTP will come and it hydrolyze the peptide chain from the tRNA. It will hydrolyze the peptide chain from the tRNA, amino acid tRNA. Understand? With the help of releasing factor 1 and 3 along with the GTP, it will occupy the A site and it will hydrolyze the peptide chain from the peptidyl tRNA. Peptidyl tRNA. And so what happens? So, the peptide chain, the tRNA, the empty tRNA, the 40S, it get dissociated to 40S and 60S subunit, releasing the releasing factor GDP and inorganic phosphate. That is the termination phase. Okay, so better take down the figure also. So that is termination phase. So what happens during the termination phase? If a stop codon comes, there is no amino acid tRNA to recognize the stop codon. So what happens? The releasing factor along with the releasing factor 1 along with factor 3 and GTP will hydrolyze the peptide chain from the peptidyl tRNA and dissociating 40S, the 80S subunit to 40S, 60S subunit and tRNA ready to carry on the next protein synthesizing phase. Is it clear? Let's take down the figure also. So that this figure shows the termination phase of protein synthesis. So this is the 80S subunit. We can see that the stop codon comes for releasing factor along with the GTP. 1 and 2 along with the GTP will come and occupy the amino acid to the A site, occupies the A site. The next phase, it will hydrolyze with the help of a molecule of water. If the peptide chain is being removed from the peptidyl tRNA. Upon the peptide chain, this is the elongating peptide chain with tRNA 40S, 60S subunit, and the releasing factors are being released. Are you able to take down the figure? Okay. So that is the termination. കഴിഞ്ഞില്ല. Yes, കഴിഞ്ഞല്ലോ അല്ലേ? ക്ലാസ്സിൽ 74 സ്റ്റുഡൻസ് ആണ് ഉള്ളത് അല്ലേ? ആരും ക്ലാസ്സിൽ കയറുന്നില്ല. Yes, കഴിഞ്ഞല്ലോ. Now coming to, so that is about the process of translation. Now coming to inhibitors of translation. Inhibitors of translation. So when the question is being asked, you have to write translation, inhibitors of translation and post-translation modifications. അങ്ങനെയായിരിക്കും essays ചോദിക്കുന്നത്. Now coming to inhibitors. Inhibitors can be bacterial inhibitors as well as mammalian inhibitors. Mammalian inhibitors. Inhibitors of protein synthesis. See, most of the bacterial inhibitors are acting as antibiotics. Most of the antibiotics inhibits bacterial protein synthesis. Most of the antibiotics inhibit the action of most of the antibiotics is by inhibiting bacterial protein synthesis. Understand? So this, you all know, the antibiotics are being synthesized from microorganisms. And these bacterial antibiotics are in turn being classified into irreversible inhibitors and reversible inhibitors, have been classified into irreversible inhibitors and reversible inhibitors. Irreversible and irreversible inhibitors and reversible inhibitors. Reversible and irreversible inhibitors. Irreversible inhibitors are bactericidal. Irreversible inhibitors are bactericidal. What do you mean by bactericidal? That means it kills bacteria. It kills bacteria. So these antibiotics are acting on which ribosome? It is the 70S ribosomes. Antibiotics acts on, inhibits the 70S ribosomes. Bacterial ribosomes are 70S, whereas eukaryotic ribosomes are 80S ribosomes. So this 70S ribosome, two subunit edekayana, 50S and 30S subunits. So the irreversible inhibitors include streptomycin. Streptomycin, irreversible inhibitors include streptomycin, neomycin, and gentamicin. So these are the irreversible inhibitors which are acting as bactericidal agents. So how does this act? They will change the conformation of the 30S ribosomal subunit of prokaryotic ribosome. They will change the conformation of the 30S ribosomes. Will change the conformation of 30S ribosome. So the mRNA leads to misreading of mRNA. Leads to misreading of mRNA. Understand? Misreading of mRNA. So what is the action of the this irreversible inhibitors? Irreversible inhibitors are streptomycin, neomycin, and gentamicin. They change the conformation. They change the protein structure of 30S ribosomes. So the mRNA can't be read. So it leads to misreading of mRNA and thus inhibits protein synthesis. Understand? So that is the action of irreversible inhibitors, mainly streptomycin, neomycin, and gentamicin. Now coming to the reversible inhibitors. Reversible inhibitors. So the reversible inhibitors include tetracycline. Tetracycline. So how does tetracycline act? Tetracycline will bind to the larger subunit. It will bind to, it will bind to the 30S ribosomes. It will bind to the 30S ribosomes and it will inhibit the binding of aminoacyl tRNA to A site. How does tetracycline act? By inhibiting the binding of aminoacyl tRNA to A site. Inhibits the binding of aminoacyl tRNA to A site. Understand? They act on the 30S subunit. How does it act? Inhibits the binding of aminoacyl tRNA to A site. So that is the action of tetracycline. Now comes erythromycin and clindamycin. Erythromycin and clindamycin. Clindamycin. How does it act? It acts at the 50S ribosomes. It acts on the 50S ribosome, 50S subunit of. How does it act? It will interfere with the translocation process. Erythromycin and clindamycin interfere with the translocation process. It will interfere with the translocation process. Understand? Erythromycin and clindamycin interfere the translocation process of elongation process in protein biosynthesis. It acts at the 50S subunit. Now, chloramphenicol. Chloramphenicol. How does it act? It will inhibit the action of peptidyl transferase. It will inhibit the action of peptidyl transferase. So these are the reversible inhibitors. Which are the reversible inhibitors? Tetracycline, erythromycin, clindamycin, and chloramphenicol. What is the action of tetracycline? They will inhibit the binding of amino acid tRNA to A site, and erythromycin and clindamycin will inhibit the translocation process, translocation of peptidyl, the amino acid tRNA to P site. Understand? It will inhibit the translocation. That is the movement of peptide amino acid tRNA or the peptidyl tRNA from the A site to P site. And chloramphenicol inhibit the peptidyl transferase enzyme. So which is a ribosomal subunit in bacterial peptidyl transferase? It is 23S ribosomal subunit. Understand? 23S ribosomal subunit. Whereas in eukaryote, it is 28S ribosomal subunit. Here it is 23S ribosomal subunit in it. Eukaryotes, it is 28S ribosomal. Here it is 23S ribosomal subunit. So these are the reversible inhibitors. So these are the inhibitors in bacteria. Now coming to the inhibitors of mammalian inhibitors. Mammalian inhibitors. Mammalian inhibitors are not used as in clinics. They are clinical. They are used in as a research tool or during clinical trials. So they include puromycin. This puromycin has got structural similarity to tyrosine amino acid tRNA. Tyrosine amino acid tRNA. So it has got, puromycin has got structural similarity to tyrosine amino acid tRNA. So what happens? Instead of tyrosine entering to the A site, the puromycin will enter and it will inhibit the protein synthesis. And this puromycin can act both on eukaryotes and prokaryotes. Understand? So that is puromycin. The next one is cyclohexamide. Cyclohexamide. So it inhibits peptidyl transferase. Peptidyl transferase of eukaryotes. Peptidyl transferase of eukaryotes. That is cyclohexamide. Next is diphtheria toxin. Diphtheria toxin. So it is a toxin from foreign bacterium diphtheria that is located in, usually seen in the respiratory tract of infected patients. So what is the action of diphtheria toxin? Diphtheria toxin inactivates eukaryotic elongation factor 2. Eukaryotic elongation factor 2. Understand? It will inhibit eukaryotic elongation factor 2. That is the action of diphtheria toxin. Diphtheria toxin being secreted from foreign bacterium diphtheria and it is located in the respiratory tract of infected patients. So that is the action of diphtheria toxin. Now comes ricin. Ricin from castor beans. So what is the action of ricin? Ricin will inhibit peptidyl transferase enzyme. Again, it will inhibit peptidyl transferase enzyme. So these are the mammalian inhibitors. Which are the mammalian inhibitors? Puromycin, cyclohexamide, diphtheria toxin, and ricin. So if you are asked to write the short notes on inhibitors of protein synthesis, you have to mention bacterial inhibitors and mammalian inhibitors. Under bacterial inhibitors comes reversible inhibitors and irreversible inhibitors. Irreversible inhibitors are streptomycin, neomycin, and gentamicin, streptomycin, neomycin, and gentamicin. And reversible inhibitors are tetracycline, erythromycin, clindamycin, chloramphenicol. And the mammalian inhibitors include puromycin, cyclohexamide, stereotoxin, hericin. Understand? So these are the inhibitors of protein biosynthesis. Now we word about protein folding. So what happens when protein, see, you know, what are polysomes? A word about polysomes. Let's see the mRNA. So, a protein, this mRNA is being translated by a number of different ribosomes, and collection of these ribosomes forms polysomes. Understand? Collection of these ribosomes form polysomes. Understand? They are located some 34 amino acids apart, 34 nucleotides apart. Understand? These are polysomes. You know, the proteins after synthesis, what happens? They have to be, they undergo organization. Proteins will undergo organization by folding. By folding. Protein folding. I'm not going into the details of protein folding. Proteins after synthesis will undergo higher order organization. So it will attain secondary structure, tertiary and quaternary structure by protein folding. And these are being mediated by mainly two enzymes, disulfide isomerase, and cis-trans isomerase. Cis-trans isomerase are the two enzymes that are necessary for protein folding. And this protein is being, and it is the chaperones which provide the environment for protein folding. Chaperones will provide the environment for protein folding, and the main chaperones that are involved are the heat shock proteins, and chaperonins. Heat shock proteins and chaperonins are involved in protein folding. And the defect in protein folding will lead to prion disease. Defect in protein folding will lead to prion disease or spongiform encephalopathy. It affects mainly brain, causing holes, riddles in brain. Brain will be riddled with holes. Or otherwise, it is termed spongiform encephalopathy. And the diseases coming under prions include Creutzfeldt-Jakob disease, and the important one is Alzheimer's disease. The important one is Alzheimer's disease. Is due to defect in protein folding. So what happens here? Mainly beta amyloid bodies are getting deposited in mainly hypothalamus and basal ganglia region. Hypothalamus and basal ganglia region, leading to dementia. See, in CT scan finding is beta amyloid bodies in hypothalamus and basal ganglia. So, so defect in protein folding leads to prion disease. Now, moving on to the next important topic, that is post-translational modification. Post-translational modification.
Ma'am, Alzheimer's മുമ്പ് പറഞ്ഞ disease.
Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease. If you have interest in reading prion disease, you can have a you can refer Devlin, the textbook of Devlin. Devlin textbook. നിങ്ങളിപ്പം textbook ആയിരിക്കില്ല വായിക്കുന്നല്ലേ, Google search ചെയ്യുന്നു. Okay. Now moving on to the text, that is post-translation modification. See, most of the newly synthesized proteins, after both prokaryotic and eukaryotic translation, they are not functional. They attain a three-dimensional structure by protein folding. After folding, they undergo structural and chemical alteration. After folding, these proteins will undergo structural and chemical alteration to become functional. And these changes or the post-translational modifications include one, it is trimming. Second is covalent modifications. Then subunit aggregation. So these are the three headings coming under post-translation modification. Subunit aggregation. So post-translation modifications include trimming, covalent modification, and subunit aggregation. What do you mean by trimming? That after proteins are being synthesized, oligopeptide chains are getting removed from it. Oligopeptide chains are getting removed. See, these are the proteins that are undergoing post-translation modification. See, proinsulin to insulin, proinsulin to insulin, procollagen to collagen. This, then zymogens. So these are the three proteins which undergo post-translation modification by trimming. Short peptide chains are getting removed. Proinsulin, you know that C peptides are removed from it. Procollagen to collagen, zymogens, which are the proteolytic enzymes, trypsinogen, chymotrypsinogen, carboxypeptidases, procarboxypeptidases. This will undergo proteolytic cleavage. The short length of peptide chains are getting removed and they are being converted to the active form. So that is by trimming. Then coming to covalent modifications. Covalent modifications include gamma carboxylation. I think you have studied gamma carboxylation. Which is a vitamin. Vitamin helping in gamma carboxylation.
Vitamin K.
Vitamin K, no? Yes, this vitamin K. Vitamin K helps in gamma carboxylation, and gamma carboxylation is needed for which are proteins? Mainly the floating factor. Floating factors. So gamma carboxylation of floating factors, then phosphorylation. Phosphorylation of which all enzymes? Glycogen synthase, glycogen phosphorylase enzyme will undergo phosphorylation, making them active as well as inactive. Like glycogen phosphorylase in the phosphorylated form is active, whereas glycogen synthase in the dephosphorylated form it is active. Okay. So covalent modification include gamma carboxylation, phosphorylation, also glycosylation of proteins. Carbohydrate chains are being oligosaccharide units are being attached to proteins. These oligosaccharide units are getting attached to protein either by O-glycosidic linkage or N-glycosidic linkage. O-glycosidic linkage means the oligosaccharides are attached to the basic, the hydroxy containing amino acids such as serine and threonine. Whereas by N-glycosidic linkage, these oligosaccharides are getting attached to asparagine by N-glycosidic linkage. Understand? That is glycosylation. Glycosylation is needed for needed signaling the proteins to their distinct site. Signaling glycosylation helps the proteins to reach the distinct site. Understand? Mainly for secretory proteins as well as the proteins for degradation. See, lysosomal site proteins പോകണമെങ്കിൽ mannose 6-phosphate has to be attached. A signal has mannose 6-phosphate is a signal which helps in signaling the proteins to the lysosomes. Understand? And defect in this process, protein targeting will lead to a disease referred as I-cell disease, inclusion cell disease. Inclusion cell disease. Mannose 6-phosphate phosphorylation defective leads to I-cell disease. So these proteins can't reach the lysosomes. So what happens? It will be seen as inclusion bodies. Inclusion bodies. So defect in glycosylation leads to I-cell disease. So that is a covalent modification include gamma carboxylation, phosphorylation, glycosylation, even methylation, ADP ribosylation, histone acetylation. These are all examples for covalent modifications. Now subunit aggregation. Subunit aggregation. Immunoglobulins, you know there are subunits in it. Also in hemoglobin, you know that there are subunits in it. And these subunits are getting aggregated by post-translational modification. Understand? So these are the process coming under post-translation modification, trimming, covalent modification, and subunit aggregation. During trimming, you can give examples of insulin, proinsulin, procollagen, and zymogens are getting activated by post-translation modification by removing short length of peptide chains. Covalent modifications include gamma carboxylation, phosphorylation, glycosylation, acetylation, ADP ribosylation, methylation, etc. Whereas subunit aggregations, example, immunoglobulin and hemoglobin. The subunits are aggregated as a result of post-translational modification. So that much completes the translation process. Translation very important, can be asked as an essay for you. You have to write the steps of translation. Usually the essay question will be the steps in translation process, inhibitors of translation and post-translational modification. Understand? എനിക്ക് ഇനി 2 ക്ലാസ്സും കൂടെ മതി. Regulation of gene expression and then mutation. അതോടെ completes genetics കഴിയും. എനിക്ക് തോന്നുന്നു ഇനി നിങ്ങൾക്ക് റിമൈനിങ് ടോപ്പിക്സ് ഇൻ ബയോകെമിസ്ട്രി വന്നിട്ട് ഡീടോക്സിഫിക്കേഷൻ എടുത്തില്ലെന്ന് തോന്നുന്നു അല്ലെ? Detoxification, free radicals, then extracellular matrix, cancer chemistry. ഇത്ര ടോപ്പിക്സേ ഉള്ളൂ. ഇതോട് ബയോകെമിസ്ട്രി ക്ലാസ്സസ് കഴിയും. Yes, thank you students. Thank you all.
Thank you,
Thank
ma'am.
you, ma'am. Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
അപ്പോഴ്
Good morning, dear students. Today I will summarize the important points that has been discussed in the motor system classes. So mainly we will discuss about the stretch reflex, inverse stretch reflex, the alpha gamma co-activation, then about the gamma efferent discharge, all these things. All these things are little complicated when you first read, but when you correlate all the aspects, it is very simple to explain. So we will summarize all these points now. Now we will discuss about the gamma motor neurons and fusey motor system. Gamma motor neurons and the intrafusal fibers they innervate together called fusey motor system. Axons of gamma motor neurons terminate in one of the ending, each located distal to the sensory endings on the striated poles of spindle fibers. The nerve terminals are either plate ending or trail endings. Plate endings occur predominantly on back fibers, but trail endings are seen primarily on chain fibers and also on back fibers. So trail endings present primarily on chain fibers but also on back fibers. The plate endings of gamma motor neurons predominantly found on the back fibers. This helps independent control of the nuclear bag and nuclear chain fibers in the spindle. So, gamma motor neurons and the intrafusal fibers, they innervate together called fusee motor system. So, in this diagram, you can see the effects of various conditions on muscle spindle discharge. The uppermost diagram, you can see the muscle spindle, that is the central one. So this muscle spindle is arranged in parallel with the extrafusal fibers. They are seen on both sides of the muscle spindle. So the tendons are seen at the top portion of the muscle spindle and extrafusal fibers. So the first diagram, the muscle is at rest. So during the rest, the spindle activity, you can see in this circle the moderate activity of the muscle spindle. They are produced by the sensory nerve endings innervating the muscle spindle. Then the second diagram, it is designated as loading the spindle. Here the muscle is stretched. When the muscle is stretched, you can see the activity of the spindles. They are increased. The next diagram, you can see the unloading the spindle. Here the muscle is contracted. That means the extrafusal fibers, when they contract, there is slackening. So there is slackening of the spindle. Therefore, the spindle activity becomes almost zero. So there is no impulses coming from the afferent nerve endings of the spindles. That is when the muscle is contracted. This is known as unloading the spindle. The next diagram, you can see the increased gamma efferent discharge. So when the extrafusal muscle fibers are contracted, it will produce increased gamma efferent discharge to the muscle spindle. The next diagram shows increased gamma efferent discharge, and this will result in muscle being stretched. So here you can see the intrafusal fibers as well as extrafusal fibers, they are stretched and there is increased gamma efferent discharge. So, from the previous diagram, you can see the effect of gamma efferent discharge when the muscle is loaded and unloaded. So, after unloading of the muscle, the spindle activity, that is the afferent activity, becomes 0. Then the gamma efferent discharge increases, and this will regulate the stretch reflex. So that is the alpha gamma co-activation. So, coordination between sensory and motor neurons is important in muscle contraction. When the muscle is at rest, the muscle spindles are slightly stretched. And type 1A afferent nerves exhibit a slow rate of discharge of action potentials. So this you have seen in the first diagram. Next, contraction of the fibers from muscles increases the firing rate in type 1B afferent fibers, that is from Golgi tendon organ. So, this Golgi tendon organ, as you know, it is arranged in series with the extrafusal muscle fibers. Therefore, when the activity from the 1B endings, when it is increased, and it is connected to the extrafusal fibers through a inhibitory interneuron. So, as you know, this is inverse stretch reflex. So, reduce the inverse stretch reflex, and it will cause relaxation of the extrafusal fibers. Then, type 1A afferent fibers, when it ceases firing, the shortening of the surrounding extrafusal fibers and intrafusal fibers as well . So, type 1A afferent fibers, when they cease firing, the shortening of the surrounding extrafusal fibers as well as intrafusal fibers occurs, thereby slackening their tensions and silencing the spirally warm type 1A neurons. Load were placed on the spindle, the slack around will be if a load were placed on the spindle, the slack around will be removed and the spiral endings would resume their sensitivity to stretch. So, if a load were placed on the spindle, the slack around will be removed and the spiral endings would resume their sensitivity to stretch. This role assumed by gamma motor neurons that they reload the spindle during muscle contraction by contracting the ends of spindle fibers to stretch the type 1A afferent endings in the middle. So they fire simultaneously with alpha motor neuron units and the muscle will be contracted. Now, alpha gamma co-activation. During voluntary muscle contraction, there is a linear relationship between spindle activity and strength of contraction. The recruitment threshold for a given spindle afferent depend on its fusimotor innervation, mainly the desired muscle force rather than on muscle length. So, the recruitment threshold for a given spindle afferent, it will depend on its fusimotor innervation. That is, the muscle spindles. Muscle spindles, as you know, they are having the afferent nerves. They are the 1A afferents and group 2 afferents. And as you know, they are having motor innervation by the gamma motor neurons. And also the fusimotor system, it includes the alpha motor neurons that innervate the extrafusal fibers of the same muscle. So the recruitment threshold for a given spindle afferent depend on its fusimotor innervation, mainly the desired muscle force rather than on muscle length. So, during voluntary muscle contraction, the linear relationship between spindle activity and strength of contraction. So, this is mediated by alpha gamma co-activation. So, alpha motor neurons, they supply the extrafusal fibers, whereas the gamma motor neurons, they will they have the motor supply of the spindles itself. So alpha gamma co-activation, it will mediate the spindle activity and strength of contraction during a muscle contraction. So this diagram shows the innervation, showing mainly stretch reflex pathway. So you can see the 1A fibers starting from the muscle spindle, pass through the dorsal root ganglion and it synapses in the anterior horn cell with the alpha motor neuron, which supplies the extrafusal fibers of the same muscle. This produces the stretch reflex. Then the inverse stretch reflex is also shown in the same diagram. You can see the 1B fiber from the Golgi tendon organ. That is the receptor for the inverse stretch reflex. It passes through the dorsal root ganglion and through an interneuron releasing inhibitory mediator, usually glycine. It will synapse with the alpha motor neurons present in the anterior horn cell, which supply the extrafusal muscle fibers. So there is a inhibitory interneuron, so it produces relaxation of the muscle. So this is the inverse stretch reflex pathway. Muscle spindles and Golgi tendon organs. Muscle spindle provide information about the length of the muscle and velocity at which the muscle is being stretched. Golgi tendon organs provide information about tension. Spindles are located in the body of the muscle in parallel with the main muscle fibers. Tendon organs are located in the body of muscle in parallel with the main muscle fibers. Tendon organs are located at the border between tendons and muscles in series with the muscle fibers. So this diagram of the muscle spindle shows the details of the nuclear bag fibers and nuclear chain fibers present inside the muscle spindle. On the right side, you can see the intrafusal muscle fibers in the middle, that is surrounded by a grey colored capsule. The afferent axons and efferent axons enter the muscle spindle through the capsule. The efferent axons that pierce the capsule, they send the gamma motor endings which supply the ends of the muscle spindle, that are intrafusal fibers. The afferent axons, they give sensory endings to the nuclear bag fibers that supply the contractile middle and non-contractile middle of the muscle spindle. So the next diagram, it shows the nuclear bag fibers and nuclear chain fibers. The nuclear bag fibers, there are two types. One is dynamic nuclear bag fiber and next is static nuclear bag fiber. The 1A afferent innervate dynamic and static nuclear bag fiber, that is the central portion. They also innervate the nuclear chain fibers, whereas the type 2 fibers, they innervate the static nuclear bag fiber and also the nuclear chain fibers. Here on the left side, you can see the nerve endings that innervate the nuclear bag and nuclear chain fibers. So as you know, the nuclear bag fiber, it has a globular middle portion, and the middle portion is innervated by primary afferent. It is also called group 1A fiber. So group 1A fiber innervate the globular middle portion of the nuclear bag as well as the middle portion of the nuclear chain fibers. Now coming to the gamma efferents. The gamma efferents innervating the muscle spindle, there are two types. One is static gamma efferent that innervate the non-contractile middle portion of the bag fiber, and they also send their trail endings to the nuclear chain fibers. So that is about the static gamma efferents. Now dynamic gamma efferent fibers, they have their plate endings on the contractile ends of the nuclear bag fiber. Next is group 2 fiber. They are also called secondary afferent. They will innervate the nuclear chain fibers. That is about the innervation in detail of both nuclear bag and chain fibers. Here you can see the spindle, also called muscle spindle. You know, the spindle is located in the central part of the muscle. The outer portion of the spindle, it is covered by extrafusal muscle fibers. So you can see the intrafusal fibers and extrafusal fibers. The spindle consists of the intrafusal fibers. The intrafusal fibers, as you know, are two types. One is nuclear bag and the other is nuclear chain fibers. The intrafusal fibers, they are attached to the extrafusal fibers at their contractile ends. Now coming to the innervation of the muscle spindle. So the muscle spindle, it has a contractile middle, non-contractile middle, and also contractile ends. The primary endings, that is 1A and 2 fibers, 1A and 2 sensory fibers, they will innervate the contractile middle portion of the muscle spindle. The 1A fibers, they are 17 micrometer in diameter. The sensory 2 afferents, they are 8 micrometers in diameter. These two fibers, they terminate by secondary ending in the contractile middle of the muscle spindle. Now coming to the motor innervation, it is by alpha and gamma motor neurons. The non-contractile middle of the muscle spindle are supplied by gamma motor neurons. They are 5 micrometer in diameter. The alpha motor neurons innervate the extrafusal muscle fibers. They are 14 micrometers in diameter. Now, the gamma motor neurons, they have flower spray ending, whereas the primary endings, that is 1A afferent, they have plate endings. That is about the innervation of the muscle spindle. So, this diagram shows the Golgi tendon organ. The first picture you can see the nerve fibers. They are 1B fibers. They are 16 micrometers and they will innervate the Golgi tendon organs. So you can see the Golgi tendon organs located in the muscle. On the right side, you can see the pathway through which the inverse stretch reflex or Golgi tendon reflex that is occurring. So here you can see the Golgi tendon organs. They are located near the tendons of the muscle. So these Golgi tendon organs, they are arranged in series with the extrafusal muscle fibers, and the 1B fibers, they are the sensory neurons. They will reach the spinal cord and these 1B fibers, it will cause relaxation of the extrafusal muscle fibers when the stretching is beyond a certain limit. So this is the pathway for inverse stretch reflex. In our curriculum, now we are giving more importance to the clinical aspects. So you should know the abnormalities. The most common abnormality regarding the nervous system, they are the paralysis of the limbs. Usually it may be upper limb or lower limb. So the paralysis may be due to two types of lesions. One is upper motor neuron lesion. That means the lesion in the neural tract. It is located in the pathway between cortex and anterior horn cell of the spinal cord. So that is corticobulbar or corticospinal pathway. So that is what is meant by UN1 lesion or upper motor lesion that leads to paralysis of or paralysis of muscles. Then what is meant by LMN lesion? That is lower motor neuron lesion. Here the lesion occurs in the lower motor neuron that starts from the anterior horn cell of the spinal cord, and the lesion may be anywhere from the anterior horn cell to the termination of the alpha motor neuron in the muscle it supplies. So now it's clear what is UMN lesion, what is LMN lesion. So now we just see the difference between how it can be shown in a tabular form, the difference between UMN lesion and LMN lesion. Number one, UMN lesion, it is due to lesion of the upper motor neuron that influence the activity of lower motor neuron. And second, about the UMN lesion, voluntary activity is lost, but reflex activity preserved. The third is spastic paralysis due to hypertonia, clasp knife phenomenon, and clonus can be elicited. All these terms you will study later. And the fourth is reflex arc is intact. As you know, the reflex arc that is that is only up to the spinal cord level, reflex arc. So that is something concerned with the lower motor neuron. So UMN lesions, there is nothing happening to the reflex arc. So the reflex arc is intact in UMN lesion. Then fifth point, deep reflexes are exaggerated due to increase in muscle tone. Some of the superficial reflexes are lost and some altered. Example, extensor plantar reflex. So deep reflexes, they are also called tendon reflexes, knee jerk, ankle jerk, etc. All these are exaggerated due to increase in muscle tone. Some of the superficial reflexes are lost and some are altered. For example, extensor plantar reflex. The sixth point, since nerve supply to the muscle is intact, muscle atrophy occurs slowly. So, the lower motor neuron that is supplying the muscle, that is intact. So the atrophy of the muscle is occurring very slowly. Then seventh point, reaction of degeneration and So muscle and nerve, you must have studied about reaction of degeneration. So in UMN lesions, this reaction of degeneration is absent. Then 8th point, positive Babinski sign is elicited. So Babinski sign, positive means the plantar reflex is positive. The next is EMG. EMG is normal. EMG also you must have studied in nerve muscle physiology, that is electromyogram, that is normal in UMN lesions. Next is LMN lesion. What are the differences from UMN lesion? Due to lesion of LMN in the motocranial nerve, nuclei or spinal cord that directly innervate skeletal muscle, both voluntary and reflex activity is lost. Because lower motor neuron, it is the final common part, both voluntary and reflex activities depend on the intactness of the lower motor neuron. So both are lost in LMN. Next is flaccid paralysis due to hypotonia and loss of innervation of muscle. So that is a feature of the lower motor neuron lesion. Next is integrity. So reflex arm, it mainly depends on the lower motor neuron. So it is lost in LMN lesion. Both superficial and deep reflexes are absent at the level of lesion since reflex arc is disrupted. Next, rapid muscular atrophy and muscle wasting occurs within 2 to 3 days. Then, reaction of degeneration present. So, reaction of degeneration you can refer in muscle and nerve. Then next is Babinski sign not elicited since plantar reflex absent. Then electromyogram shows fibrillations. So you know the difference between fibrillation and fasciculation. Okay. So you refer back the difference between fibrillation and fasciculation, and electromyogram in LMN lesion shows fibrillations. So that is the main difference between UMN and LMN lesions. You have to differentiate once you see a person with paralysis whether he has a UMN lesion or a LMN lesion. That's all.
ഒന്ന് മാഡം ചോദിക്കാമോ?
So now we will discuss the important questions. I will discuss only the motor system. So you prepare notes about these questions, and you will be having the assignment to be submitted on 11th. So 11th morning. I think there is some confusion since only 100 persons can join this Zoom about the attendance. So the last 4 classes attendance we will take from the assignment submitted on 11th. These questions you prepare as an assignment and submit on 11th morning. Okay. So the questions are pyramidal pathway, functions of pyramidal system, name cranial nerves originating from brain stem, differentiate UMN and LMN lesions. So that is the first question. Then second question, reflex action, classify reflexes, properties of reflexes, then examples of each type of reflex, then reciprocal innervation, final common path. So you prepare a note about these aspects. Then third question, differentiate stretch reflex and inverse stretch reflex, then function of gamma motor neuron, then what is Jedrassik's maneuver. All this I have described in the classes. So you prepare as an assignment these questions and submit it on 11th. So it can be taken as your attendance. Next is diagram of motor homunculus and write briefly on cortical plasticity. So about cortical plasticity, you have to refer little bit. I have not mentioned much, and you prepare notes on this. Then fifth question, name the extrapyramidal pathways. Then last question, what is crossed hemiplegia? So all these 6 questions you prepare and submit on 11th morning. Okay. Diagrams. So diagram book, you will be given a list of diagrams. So some important diagrams we will give as a PDF. Others we will give the list. So it will be sent by WhatsApp later. So I have told your representative, and he told that you will submit all the diagrams regarding the CNS on 18th morning. Okay. So by 1 or 2 days we will give the PDF and also the list of diagrams by WhatsApp. Thank you.
Thank you.